So yes, indeed. So I'm very happy to participate to this seminar series. Although I'm not doing, I used to work with photothermal microscopy, but I'm not doing that anymore. So you will see. So, but I will try to entertain you with some spectroscopy and applications of carbon nanotube in the context also of super resolution microscopy. To motivate a little bit this work, let me introduce actually we have a large goal in this project, which is to try to decipher or understand how molecules diffuse, move, are sequestered in a complex environment, and that could be, and you will see a biological environment, but you can see that as a more general structure actually. And for that, what we are doing is to adapt and develop single particle tracking methods, which are based very often on infrared microscopy and based on photoluminescent nanoprobes. And for that, of course, we need to design and adapt the photophysical properties of this object. And you will see that I will discuss all these strategies towards one application that we are very motivated, which is in neuroscience. So if I rephrase a little bit the motivation of this work, actually, and I will start from the bio motivation, and you will see that we go more and more into physics along the line. People do the opposite most of the time, but I find it interesting to do it this way. So in the context of a large ERC project with colleagues in neuroscience, actually, we want to understand a particular environment, which is a brain extracellular space, which is a space in between cells. And this space actually represents a fifth of the volume of our brain. It has been totally underseen up to now. And but we know that it's a very important object because because that's where you have nutrients, that's where you have hormones, where you have signaling molecules, that they all transfer to this space. And we know that the brain needs and the brain adapts its morphology, the dimensions, the chemistry inside this space to veiculate, to let the molecule diffuse, or to keep some molecule at certain places into this space. And the problem is that this dimension, the dimension of this space, are nanometric, which makes it extremely difficult to study. And if I extend that a little bit, the parameters that regulate actually these dimensions and the mobility of molecule in this space, of course, there is a geometry which is extremely difficult to understand. And that's something that we'll want to look at. But also you have some chemistry, some viscosity effects, and you have plenty of molecules into that. And we want to untangle actually how this space is functioning into the brain. And for that, we'll do single particle tracking. So there's been quite a nice, it was the cover of Physics Today about this space last year. So if you're interested to that, you can read that. And the first question that we had was just simply the structure and dimension of this space. And it was never measured in intact brain. The knowledge that people had was mainly related to electron microscopy and cryofix electron microscopy. And then, of course, the local diffusion properties of the molecule into this space were unknown. So if I want to rephrase that in a very simplified picture, the question that we have is the brain extracellular space organized in one of the structures, of course, at the nanoscale, and how molecules navigate into such structures at these levels. So our strategy will be based on single molecule detection, and I think I don't have to introduce the superlocalization properties of single emitters, and we'll use that a lot in our applications. But what I would like to highlight here is that for me, I like to separate the two main applications and two main benefits of the superlocalization of single emitter into two categories. One which is based on the diffusion properties, and people, it's been more than 30 years that people do single particle tracking. where you can have a lot of information, dynamic information, and of course, more recently, over the last 20 years still, a single molecule localization microscopy where you get structural information, and we have worked a lot in this domain over the years. And what we'll want to do actually is to benefit from the two in one single experiment, and I will explain you why I'm saying that. And actually, our strategy was the following when we started this project. We were saying, okay, imagine that you can put a single molecule into the brain extracellular space. Imagine that this molecule is bright enough that you can detect it at depth into a tissue. Of course, we need to be inside an intact tissue to do that. Imagine that the molecule can navigate efficiently enough so that it can go in narrow spaces, but at the same time give a lot of data points. So it means that you have a very efficient exploration. Then, if you concatenate all the position of these molecules or this emitter, you can have a super resolved map. So it's the structural information about the space that you're looking at. And at the same time, you also, if you analyze the diffusion properties, you have the local diffusion properties into this space. And it turns out that to perform all of this all together, and we have studied on many types of nano objects, the carbon nanotubes open this possibility, because you will see that they are in the shortwave infrared emitters. They have efficient space exploration. I will show you that, and absolutely exquisite photostability. So in the world, I would say, of SWIR emitting particles, you know that in biology, people are very motivated to generate new types of fluorophores in the infrared, and I list a few of them here. And this nice review by the group of Umjidai, which is a bit dated now, but still valid. I want to introduce here for you the single-wall carbon nanotubes for single molecule applications. So we are talking about single-wall nanotube, meaning that you can see them as a sheet of graphene that you roll to create such structures. The diameter will be around 1 nanometer, the length between tenths of nanometer up to microns. And you can show that depending on how you roll the sheet of graphite here to create your nanotube, you can have very different characteristics. And 1/3 of these structures are metallics, and the 2/3 are semiconductors. And so I will focus on the semiconductor ones here. And you can show that each chirality, so we call them chirality, is structure actually has distinct photophysical properties. Now it's possible to separate them very efficiently. So you see you can have many types of fluorophores. And you see here absorption spectra of each of these type of chiralities. And then when you'll see here, there is a band edge exciton here, which is this one. This is a second order exciton here. And you see that the emission is in the near infrared. And in 2002, the group of Bruce Wiseman demonstrated the photoluminescence and bright photoluminescence of such nanotubes. So here is a 2D photoluminescence map where you have emission wavelengths here, excitation wavelengths here. And if you pick, for instance, this one, which emit at 1100 nanometer, excited at 650, actually, these are 76 nanotubes, which are somewhere here, which are those nanotubes here. And so each peak corresponds to one type of nanotube. So it provides very interesting properties. So over the years, actually, there's been the golden years of the spectroscopy of single-wall carbon nanotubes between, I would say, 2005 and 2015, where we and others, of course, performed a lot of spectroscopy of such carbon nanotubes at the level of the single tube and understanding excitonic processes in this for these objects. And then later on, of course, the main applications that were coming in terms of the optical properties of carbon nanotubes, and I would separate them in two classes. The one with quantum light sources, so you can do single photon sources with carbon nanotubes. And the good thing is that they emit in the near infrared, so in the telecom band. So that's something interesting. And obviously, also bioimaging is something interesting because you see that the luminescence properties fall in the transparency windows of the tissues. So I promised just a little bit of photothermal. Of course, over the years, we looked at the photothermal properties of this of the carbon nanotubes because they are very strong absorbers, and so they are they provide very high photothermal contrast. So over the years, that's some of the papers we published along the line. So 2007 to 2013, some spectroscopy of carbon nanotubes. So these are semiconducting. Here, these are metallic carbon nanotubes. We did by combining photoluminescence, photothermal, and also modulated absorption. We could measure the absorption cross-section extremely precisely about these carbon nanotubes. And then some imaging applications here. It was a measurement of ultra-short carbon nanotubes. You will see that I will come back to that. So these are photothermal images of ultra-short carbon nanotubes done to dimensions which are around 15 nanometers in length. And also some bio applications here where we look at the degradation of some carbon nanotubes by primary cell, phagocytic cell, that actually we see the degradation of the nanotubes by the same cells themselves. Sorry. Okay, so to come back to the application that I was, so come back to photoluminescence properties here, I would like just to highlight the diffusion properties of these carbon nanotubes in a complex environment. So the first thing that I would like to show you is the example of fluorescent beads, 20 nanometer fluorescent beads in an agarose gel that we can see as a model system. And you see that they diffuse extremely efficiently. So in such a system, so you can say, okay, this one would be interesting. But the problem is that they diffuse too fast and they don't provide a lot of data points, and you cannot retrieve such informations like this. And now if you take nanotubes on the contrary, actually, so this is a very long nanotube. This is a shorter nanotubes here. And we discovered in 2010, actually, that when you have very long nanotubes, due to some flexibility, they can rotate, diffuse into such environment extremely actually efficiently. They provide a lot of data points. You see, they follow the canals like this. So this movie is 20 minutes. You see that the photo stability of this object is very interesting. But this is, okay, this was interesting, but this is not the situation that we want for bioimaging. And actually, in between spherical particle and very long 1D objects, actually, we have, let's say, a sweet spot here, nanotubes which are around 500 nanometer to 1 micron. And you see that we are exactly in this regime. And really, the point that to understand here is that the small dimension of the nanotube allows them to go in very narrow environment, like the agarose, like the beads, but also their lengths make them bump into the corner, I would say, the boundaries, which provides a lot of data points, and you see it by eye, actually, that you have a lot of points. And that's exactly what we want for our applications. You see also that there is no photo bleaching. I mean, they are absolutely photostable. They are very bright. And we are talking about near-infrared emission. So we worked then on these carbon nanotubes to make them biocompatible. So I will not discuss that, but that was a lot of work also to make them stealth, so that there is no non-specific interactions with the cells. So we did that. And also we did some spectroscopy where we looked in terms of excitation of the nanotube, because the photophysics is extremely rich. What we could, the different strategies that we could have, so either up conversion, excitation on a second band, or excitation on a phonon side band. And it turns out that the most efficient in terms of energy deposition in the system, signal-to-noise ratio, etc., was to use the phonon side band excitation. So that's what we use in our applications now. So then there was some bio parts. So we designed strategies to inject the carbon nanotubes into the brain of mice or rats. So what we do is that we inject in vivo, meaning that we do that on the live animal. We inject them in the ventricle of the brain, and we let the biology actually of the brain do the job, where you have a very efficient dispersion of the carbon nanotubes all over the brain. So that was very interesting. We saw that. And then after half an hour, we terminate the animal, we prepare the brain, and we create brain slices. So it's acute brain slices that we put on our microscope. And this microscope is a 1 photon wide field microscope. So nothing fancy, no confocal, no multiphoton. So we are exactly like this. And we are able to detect the movement of individual carbon nanotubes like this, and really at depth. I mean, for single molecule imaging, you can imagine that we are really deep into the brain tissue, and you see that it works pretty well. So this was an experiment with a lot of nanotubes. Of course, we work with more diluted samples. And from that, actually, you can generate some trajectories, exactly the way I told you. And then we can create a map, a 2D map here, of the position where the molecules are. And you see that here from 20,000 localizations, so it's a very long trajectories that provide a lot of information, we can generate a super resolved map of the extrasolar space architecture. And the third thing, of course, we did was to compare that to cryofixation. And it turns out that, I will not go into the detail how we extract the local dimension, but we developed some strategies from such map to measure precisely the local dimension. And you see that the dimension that we find are below 200 nanometers, and that they correlate very well with what we see in cryofixation. So it indicates you that actually the carbon nanotubes are very efficient explorer and very efficient probes of their environment. Then, of course, we have more than just the maps. We have also the diffusion properties, and so we are still working on that because it's very complex system, and to do some segmentation of the local region. But just a glimpse of what we are doing now is to analyze locally the diffusion properties and to correlate that to the dimensions of the extrasolar space. So we can have diffusivity maps, and if we are smart enough, maybe one day we can even come back to the viscosity, local viscosity. So here I would call more effective viscosity, because you have plenty of parameters to take into account. So just to show you a few examples of what we do with that, I will show you two quick applications. One question that we had was to look at what's going on near synapses. So this is the hippocampus of a brain. Here you see the dimension. If you zoom here in this region, you start to see the cells. And if you zoom here, you can have synapses. And what we did is we performed some correlative microscopy, so on live brain slices, where we labeled the synapses with genetic tools. And we use what is called the halo microscopy, which was developed by Jérôme Mertz some years ago, which makes some sectioning to be able to locate the synapses. And when you have that, you can look at the diffusion properties of the carbon nanotubes around synapses. And to go directly to the results, so here in the background, you see a synapse. And we were able to segment actually our trajectories, the dimensions, so that's a local dimension of the brain space, and here is the diffusivity of the molecule, the nanotubes around the synapse. And what we show by analyzing the properties of the diffusivity as a function of the center from the synapse, and same thing with the local dimension, we could see that within the 500 nanometers around the synapse, actually diffusivities are larger, and dimensions of the brain extra space are larger. And that was very clear and extremely kind of surprising. It means that in the brain, the diffusivity for molecules around in the 500 nanometer around synapses is larger. And we have seen that, I will not go into the detail, that this is regulated by synaptic activity. Another application, I will go fast on this one, is more integrated subject, but we also looked at the progression of the Parkinson disease into some brains. And to make a long story short, we have seen that there are modifications in the brain extrasolar space in Parkinson brains. And we revealed that the diffusivity and dimension are changed. And this is also related to some inflammatory response of the brains. So I will not go into much detail on that, because I have more interesting things to tell you for this audience now. So now, in the time remaining, I would like to discuss how we can augment the photophysics of these carbon nanotubes for localization microscopy in the near infrared. And all of that comes from really a new effort that has been performed in the community, which is the possibility to add some chemical function onto the nanotubes that, and you will see, so people call them defects on carbon nanotubes, and they will create actually new excitonic centers where you can have luminescence at local luminescence at these defects, and so it triggered a whole new possibilities in the field of carbon nanotubes. I would not compare that totally with NV centers, for instance, in diamonds, but it's a little bit the idea that you introduce some color centers onto carbon nanotubes. And I will show you 3 examples of what we can do with that, starting with the first one, which is how we can improve bioimaging with such color centers. And why is that? Actually, so remember, that's what I presented you. You have pristine nanotubes, regular nanotubes. You have excitation on the phonon side band, as I told you, on emission from the band gap. And when you introduce these color centers here, what happens is that actually you create a low energy states from which you have emission at the color centers here. And when you think about that, this provides you a possibility actually to perform resonant excitation on the band edge here. And that's very interesting because this is a strong, very strong, of course, absorption line. And what we did is to look at the single molecule level, so the photoluminescence intensity as a function of excitation intensity. And what we do here is that we record it by excitation at the phonon side band. So this is these lines here, with or without color centers. Everything is the same. And of course, you can obtain the exact same photoluminescence properties, but reducing by a factor of 20 the excitation intensity. And it's because simply this line is very strong. And visually, you can see that we looked at that into brain structure. So this is the same area on the brain structure where we excited here on the phonon side band, or we excited here the nanotubes at the E11 transition. But when you see here, we excited them with only 100 watts per square centimeter, which is very low in bioimaging of single molecules, with an improved signal-to-noise ratio. So it allows us now really to reduce the dose of excitation that we put into the brain, and at the same time increasing the signal-to-noise ratio. So we have a double benefit, and this is something that we are doing quite a lot now. So it's an excitation 985 nanometer and emission at 1160 nanometer. So it's really near infrared emission and excitation. So that's very beneficial. So the second thing that I would like to show you with these organic color centers is the possibility to have nanotubes which are shorter in length. Because and I will explain why, all the nanotubes that I showed you up to now had lengths typically larger than 100 nanometers, so around 500 nanometers. But it would be very interesting, of course, for different applications, of course, for the diffusion of carbon nanotubes, but also for immunolabeling, if you want to use the nanotubes as immunolabels, to have them much shorter and down to maybe 15, the size of an antibody would be very interesting. And the reason why we cannot have ultra-short nanotubes that are photoluminescent is because excitons in carbon nanotubes diffuse along the nanotubes, and the length of diffusion is around 100 nanometers. And imagine that you have a nanotube which is shorter than this diffusion length of excitons. Actually, what happens is that the ends of the nanotubes act as quenchers of the photoluminescence. And if you produce nanotubes which are, let's say, 40 nanometers, and that's the example of this sample here, you don't have any photoluminescence. And the idea now is that if you introduce this color center, what they will do is that they will trap the excitons at these color centers and allow you to retrieve now some photoluminescence. So that's exactly the same sample, but now that was functionalized with some color center, and you see very bright emission, the E11 transition that you can see here. So we could detect, so the image that we have indeed is that the excitons diffuse diffuse along the nanotube and are trapped in this defect trap, and they emit from here. We can discuss these processes as well. The good thing is that these ultra-short carbon nanotubes, actually, we can detect them at the single molecule level. That's what you see here. So emission here, 1160, median length around 40 nanometers. And the question we wanted to ask now is, can we resolve, so it was a challenge, can we resolve the emission sites along this ultra-short carbon nanotube? So we want to zoom and to do super resolution of the ultra-short carbon nanotubes here and to look at these color centers. So to do that, I'm doing a small flashback. In 2008, actually, we had introduced a strategy to do super resolution imaging of carbon nanotubes. So this is not ultra-short here, it's a regular nanotubes. And we had seen that if you create protonations onto the nanotubes, actually, it quenches locally the photoluminescence on the nanotubes. And when you have deprotonation, actually, the photoluminescence is going away. And so somehow the carbon nanotubes will flicker like this. And now imagine that when you look at a nanotube like that, which is flickering, you perform on two subsequent images, you perform the difference between those two. And actually what you see is that you can individualize the site of protonation and deprotonation. And by doing that, you can then apply the superlocalization properties to say, okay, now I can superlocalize these positions. And it works very well, and you can retrieve and make a super localization and super resolution imaging of now the carbon nanotube photoluminescence along the nanotube. So these are examples here. So this one is very short, is only 200 nanometers in length. So it worked very well. So just a small comment. I have chosen a very bad title for this article when we publish it, because there is no microscopy, no super resolution. Although it was in 2008, one of the first super resolution paper, and people have not seen it because there is not the nice keywords in it. But anyways, the work is there. And so coming back to the ultra-short carbon nanotubes now, so we adapted this method to the ultra-short carbon nanotubes, and now pay attention to the scale bar. This is 50 nanometers here, and you see that we were able now to really localize the emission centers on a single carbon nanotubes. And when you look at the distance between the different emission centers, it turned out, so we looked at this, the distribution is around 40 nanometers, and 40 nanometers actually is the distribution of length of this carbon nanotube. So this allowed us to say that the color centers are mainly at the ends of the carbon ultra-short carbon nanotubes, which is not a surprise because that's where the chemical functionalization is the most efficient. So now we're applying these ultra-short carbon nanotubes into brain imaging, and it works actually. It works pretty well. We can even access regions of the brain that longer nanotube cannot access. So we are writing the paper at the moment. But it's really interesting now to have ultra-short carbon nanotubes that emit at 1200 nanometers, and we excite them at 985 nanometers.
4 minutes left, no?
Okay, I will be done. And finally, I have 2-3 slides to show you now how we can play with carbon nanotubes to photo control their photoluminescence with certain type of chemistry. So in this case, it's another chemistry that is performed on these carbon nanotubes. The idea will not to add color centers, but is to be able to graft photoactive molecules onto the nanotube, which without changing and without perturbating the sp2 the sp2 character of the carbon nanotubes. It means that by grafting this molecule, the photoluminescence is still coming intrinsically from the nanotube. So this is work done in collaboration with Stephanie Reich and Antonio Setaro at Freie University in Berlin. And attaching, so it's really a cycloaddition here onto the nanotube. And what Stephanie and Antonio did is to add a well-known molecule for photochemists, which is spiropyran merocyanine, that has the ability to flip if you add UV onto a change conformation if you add UV on this molecule. And it turns out that if you look at the single molecule level, single nanotube level of such nanotubes, so that's what we are doing here. So we have a nanotube, different nanotubes here. Those are their photoluminescence. And when we put the UV on, what you see that the photoluminescence starts to flicker. And this comes from the fact that you have spiropiran merocyanine transition. And in the form of the merocyanine, actually, you have a quenching of the photoluminescence, a local quenching of the photoluminescence of the nanotubes. And so we looked and we analyzed the photoblinking dynamics by simulation, taking into account exciton diffusion properties, nanotube length, functional density, etc. And we can totally reproduce this type of this behavior. But what is interesting now, if you think globally, is that this behavior is exactly the behavior you want for photoactivatable, photo-switchable molecules for super resolution imaging. So we did a first experiment, very simple experiment here, where we tried to use this photoblinking to localize the carbon nanotubes like you do in Palm, for instance. So you see that you can, here you have some insets showing the super resolved image of this object. But the most interesting is here. Here you had a cluster of nanotubes that you cannot resolve, and that you see that by this Palm effect, you can resolve clearly the different nanotubes at very high precision, less than 300 nanometers. And again, here imaging, so it was another type of nanotube than the one before. It was 10.2 nanotubes, and they emit at 1064 nanometers. So we believe that we created the grounds for doing some Palm-type imaging with photoactivatable nanotubes, which can be interesting in the future for different applications. So to conclude, so that's my conclusion slide. I hope I convince you that luminescent carbon nanotubes and localization microscopy in the SWIR domain are very interesting for different applications, and especially to explore structural and the structure and the dynamics of complex topologies, and that the possibility to add organic color centers on these carbon nanotubes offer many possibilities. So I showed you for bioimaging, but you can imagine also for quantum physics. It's also something that is very interesting. So we are many perspectives in the group. Everything that I showed you was 2D imaging. Of course, we are doing the 3D now, the same thing in 3D by phase engineering. Also, the one thing that is important in the field of a single molecule is that carbon nanotubes are perfect dipoles, actually. And they are the very interesting candidates to do orientational microscopy, because the dipole is so well defined that to do to measure the orientation of diffusing molecule is something. So we are working on that. We are going towards in vivo imaging, so real in vivo imaging. So that's something, and of course some other applications in the brain and in cancer and all of that. So I just mentioned here two recent reviews that we have done. One, super resolution with carbon nanotubes 2 years ago, and also if you are interested in some of our with Pierre Bon, who was a member of my lab. What we think of the field of high-resolution optic bioimaging and where it should go. So you can raise this perspective that was a reference actually to write. And to finish, so some acknowledgements, of course. So a great team to work on this project with different students and postdocs and former students and postdocs as well. Many collaborators, so in the context of the ERC project in neuroscience, and colleagues in chemistry, and also in carbon nanotubes in France and abroad, and some fundings. And with that, I will thank you for your attention and take some question now or later. I will stay tuned.
Thank you very much, Laurent, for this very exciting talk and nice results, exciting results. I definitely have many questions, but let us see. You can ask your questions either directly or by writing them in the chat. Let me start with one before. So you mentioned that they are extremely stable, these carbon nanotubes, but yet you show that they can be degraded by the cell actually. So can you explain a bit what happens there? What is the chemistry or photochemistry that is taking place or not taking place depending on the conditions? In a few words.
Absolutely. So yeah, the example of the carbon nanotube that were degraded by the cells, actually, it's not the nanotubes that we were looking at here. It's were nanotubes. So it was a collaboration with Alberto Bianco in Strasbourg, who functionalized the nanotubes so that the nanotubes can be eaten by the cells.
So those nanotubes had been chemically modified so as to be digestible. But the normal ones, are they degradable?
No,
or?
Actually,
no. Okay,
not that I know. Not that I know.
I hear no more questions. You can raise hands or just speak.
Randy as a
Randy, please go ahead.
Great. Thanks for this really rich story. I also have a bunch of questions. The one I'll ask here is, can you comment on the difference in angle, or is there a difference in angle between the absorption transition dipole and the emission transition dipole for the chemically functionalized nanotubes?
Yeah, this is still an open question for the chemically functionalized one. Up to now, the work that I have seen was that the dipole was still along the backbone of the nanotubes, which is not obvious to me, to tell you the truth.
Not what I expected.
So there is one group who looked at that a little bit. I would like to investigate that a little bit more in detail. I don't see
particularly, I'd say particularly with your ability to look at both kind of long nanotubes like super 40 nanometers and sub 40 nanometers. Yes. You know, if you're functionalizing at the ends, you know, maybe there's sort of a different orientation of those defects. It sounds like a really interesting question.
Yeah, absolutely. That's something that I have in mind, and that's why also we are doing the orientation microscopy. You know, I'm going more and more into these questions. I don't have the answer yet.
Thank you.
I don't see any further questions, so I can just sweep another one from myself. So you mentioned, Laurent, that of course these carbon nanotubes have chirality. And so a few years ago, we have shown that by photothermal microscopy, we can investigate, you know, the left and right-handed objects. So have you plans to look at the chirality of carbon nanotubes directly by measuring, you know, with polarized light in photothermal? Yes, or other methods?
Absolutely. So this has been done, not by me, because people now have been able to even sort nanotubes by their handiness. So left chiral or right chiral, people are able to provide sample of one species or the other one for the same chirality, same structure, but left and right. And
so how do you do that? By sorting them out, or chemically?
It's chemically. It's chemically. It's your different strategy, either by DNA wrapping that selects certain types of nanotubes, and then centrifugation. You have also phase separation, two-phase aqueous separation. So there are some colleagues in the field that are absolutely expert in that, and they were able to separate them. And actually, you can show that the spectroscopy is different. So they have not exactly the same, even the same luminescence bands. You have a small shift, and people have done some circular dichroism onto that. Not me. It's not me, but some colleagues have done that.
Maybe a slightly related question. You mentioned a sample with, you know, different chirality or different photoluminescence. So are the samples sorted out according to spectroscopy or chirality, or is that
So
just by
what
chance?
Up to now, what I did up to now was to use unsorted samples, and then I select the nanotubes that are interested to me optically. But I have started now since 6 months to work with sorted samples. So that's my collaboration, because some colleagues are much more experts to do that, and to have some pure samples of one chirality.
So
I'm not talking about
chirality,
right?
but also spectroscopy, right? I mean, can you sort them out by, you know, the 7.6 or whatever?
Yeah, you can have pure 7.6. You can have pure.
So how can you do that? I don't know how to.
Again, it's some
By chemistry.
phase separation.
Nice.
So you have two families. So either phase separation, SDS, SDBS, and you have some phase separation. It works.
Nice.
Or by wrapping, selective wrapping, selective encapsulation of the nanotubes, and that's with DNA. And actually, that's the most efficient way to do it today. I showed you the spectrum, absorption spectrum, a list of absorption spectrum in one of my slides. That was done by DNA.
Done by this sorting, by chemical recognition, sort of.
Yeah. And
nice.
It's incredible. I mean, it's incredible what people can do. It works very well.
Impressive. Thanks a lot. Are there more questions from the audience? I don't see any.
I can ask one more if there's.
Please go ahead, Randy.
Thanks. So maybe this isn't so useful for biological imaging, but you know, there's always advantages for having emitters at telecom wavelengths. If you functionalize these nanotubes, again with the chemical modification, can you get emission at 1300? Can you get emission at 1500 nanometers?
Done. It's done. It's been done. It's been done, and people have done. So people do quantum optics with these objects, put them in cavities, and they have. It has been. It has been shown some single photon sources. Single photon sources has been done at this wavelength and at room temperature, even.
Thank you.
So, well, if you look at the recording of my presentation, I provided a nice review on that one. It was a few years ago by Christophe Voisin and colleagues. And you have all the state of the art, at least a few years ago, onto that one.
Great. Thanks for directing me toward that.
All right. Thank you very much. I see no further question. And anyway, you can ask more questions after the last talk of Masaru Kuno in several minutes.